All right, let's dive right into this explainer and take a really fascinating zoomed-in look at the microscopic mechanics that literally keep living organisms alive. We hear a lot about how our bodies process food, generate energy, and just, well, keep us functioning day to day. But today we're looking at the actual biological engines that make all of that possible. We're talking about enzymes. Here is our roadmap for today. We'll start with the biological catalysts, then unpack the lock and key mechanism. After that, we'll explore temperature and kinetic energy, look at the impact of pH, and finally discuss the danger of denaturation. But before we get into the heavy technical mechanics, just consider this ultimate high-stakes reality. Without these highly specialized workers, your cellular factory literally crawls to a halt. And life? It's simply impossible. They control the reactions in your cells and make absolutely sure these reactions occur fast enough for the cell to actually function. Without them, we simply don't survive. Period. All right, moving right along to Section 1, the biological catalysts, the factory workers of your cells. So the crucial point we need to nail down right away is separating the general definition of a catalyst from an enzyme. A catalyst is, broadly speaking, any substance that increases the rate of a chemical reaction without actually being changed by it. But an enzyme is specifically a protein that functions as a biological catalyst. They're involved in all sorts of metabolic reactions, like the breakdown of glucose to release energy through respiration, for example. And honestly, what's amazing about this is that just like general catalysts, these enzymes can be used repeatedly. Why? Because they are completely untouched. They aren't used up during the reaction itself. They are essentially the ultimate reusable workforce. Which brings us to Section 2, the lock and key, how enzymes actually work. Now, to really understand this microscopic factory floor, we first need to define our basic components. First up, this substrate. This is a raw material. It's the substance on which the enzyme actually works. Next is the active site. This is the incredibly specific part of the enzyme that's responsible for combining with that substrate. You can kind of think of it as a highly specialized docking station. And finally, the product. This represents the finished goods, the brand new molecules that are produced after the reaction has taken place. So, how does this all come together? Well, here is exactly how these components interact in what scientists call the lock and key model. First, the substrate approaches the active site. And because that active site has a highly specific shape, it fits the substrate perfectly, literally just like a key fitting into a lock. When they lock together, they form what's known as an enzyme-substrate complex. The enzyme essentially grabs the molecules, brings them closer together, and facilitates the chemical reaction. And boom, once the reaction is complete, the products are quickly released, which frees up the enzyme to instantly go off and catalyze even more reactions. Now, you might be wondering, why does this specific shape matter so much? Well, it is all about precision. Enzymes are incredibly specific. Because of this highly tailored active site shape, an enzyme like, say, a protease, will only ever catalyze its complementary substrate, which in this case is breaking down proteins into amino acids. It is completely 100% unable to bind to glycogen or starch because their molecular shapes just aren't complementary to the protease active site. The lock will only ever accept the correct key. No exceptions. Let's zoom out a bit for Section 3, temperature and kinetic energy, heating up the cellular factory. Okay, so when you turn up the heat in the cellular environment, it triggers a really fascinating cascade. Generally speaking, a rise in temperature increases enzyme activity. Both the enzyme and the substrate molecules gain more kinetic energy from that heat. And this extra energy means they literally move faster. They're zipping around. Because they are moving at higher speeds, the frequency of effective collisions between the enzymes and the substrates naturally goes way up. And, as you'd guess, because they're colliding more often, the overall rate of reaction and product formation rises rapidly. However, that productivity doesn't just scale up infinitely. 50 degrees Celsius represents a very dangerous breaking point. As the temperature goes up, enzyme activity continues to rise until it hits its absolute optimum level. But once we push above that 50 degrees Celsius mark, most enzymes, because they are proteins, begin to break down and they stop functioning correctly. That incredible hyperproductivity we just talked about comes to a crashing halt. And that leads right into Section 4, the impact of pH, the acidic and alkaline environments. It turns out acidic and alkaline conditions actually alter the structural properties of proteins. Because of this, most enzymes work best at a particular pH level, but this optimum state differs vastly depending on what the enzyme's job actually is. For example, let's look at pepsin, which is a protein digesting enzyme found in your stomach. It has an optimum pH of around 2, which is highly acidic. It absolutely needs that extreme acid to thrive. Now contrast that with amylase, an enzyme found in your saliva. Amylase works best in neutral conditions, right around a pH of 7. If the pH increases or drops too much from these specific optimums, enzyme activity is going to slow down and eventually stop completely. Which brings us to our final segment, Section 5, the danger of denaturation, when the factory breaks down. We need to talk about a really crucial concept here, being denatured. When an enzyme is heated too much or exposed to the wrong pH, the protein's actual structure is altered. Remember our lock and key analogy from earlier? Well, when an enzyme becomes denatured, its shape changes, which means the highly specific shape of its active site changes too. The lock is, for all intents and purposes, melted. Because of this, the enzyme-substrate complex can no longer form. The specific keys just don't fit anymore, the rate of effective collisions declines drastically, and those countless chemical reactions that keep an organism alive, they start happening entirely too slowly. Now, if you're struggling to visualize this kind of structural loss, just think of cooking an egg. When you heat an egg white up in a pan, its color and consistency completely change, right? That is denaturation happening right before your eyes. The proteins lose their original shape due to the heat, and once they change, they are altered forever. You simply can't uncook the egg. But here is a very critical distinction that you should definitely take away from this explainer. While high heat causes that permanent structural loss, just like our cooked egg, denaturation caused by extremes of pH actually operates a bit differently. Interestingly enough, the loss of function caused by extreme pH is generally temporary. That means normal function can actually resume once the pH returns to its appropriate optimal level. The enzyme recovers. And that leaves us with a fascinating thought to end our explainer on today. We know our enzymes are these incredibly specific, powerful little machines that literally dictate our survival. And we know that while they can temporarily bounce back from pH extremes, high heat breaks them down permanently. But as we continue to unlock the complex secrets of biochemistry, you really have to wonder, if normal function can resume after pH extremes, could scientists one day engineer human enzymes that completely bypass the permanent lethal rules of heat denaturation? Just how resilient could we make the human cellular factory? It's definitely something to think about the next time you consider the microscopic miracles keeping you alive right now.